Hello, today I'm going to be talking about the Virus Infectious Disease Ontology and the COVID-19 Infectious Disease Ontology and coordinating coronavirus research using these tools. Before I jump into that, do just a little bit of preliminary background, covering specifically the Infectious Disease Ontology ecosystem, and then I will hop into each of the ontologies in turn. All right, starting here, I'm going to talk a bit about the infectious disease ontology itself, as well as extensions, and then make clear the connection to the coronavirus infectious disease ontology. So, IDO Core is an extension of basic formal ontology to infectious diseases. It provides researchers terminological content, as you can see, for domains covering specific infectious diseases like brucella, influenza, and more recently coronavirus infections. The arrows there pointing from IDO Core indicating that is an extension. So IDO Brew, for instance, extends IDO Core, so does CIDO. Proliferation of virus-specific infectious disease ontologies in recent years, like CIDO, has warranted the development of a virus-specific reference ontology. This is the motivation for VIDO. Now, I will illustrate some aspects of VIDO, particularly the interoperability it exhibits with several other widely used ontologies, and then some of the core terms. Here's a lot of ontologies referenced. The color coding, as you can see over here on the right hand side. Hopefully some of these acronyms look familiar. You have BFO at the top, you have GO, gene ontology, then there's IDO Core. CL is cell ontology, and then VIDO, and then a common core ontology below VIDO, and then OBI, IO, Uberon, and then IDO COVID-19, and then CIDO here in gold. This color code is to be used here for the diagrams to the left, indicating subclass and then shortcut relations holding among these terms. I'm going to walk through some of these to illustrate aspects of VIDO. Acellular structure, what is this? So many ontologies classify viruses as instances of organism, a term imported from the OBI. But viruses are acellular, and the definition of organism strongly suggests that instances have cells. So VIDO does import organism, but it classifies viruses under an IDO Core class, acellular structure, which was developed to avoid this issue. Here's a definition: an object consisting of an arrangement of interrelated acellular parts forming an acellular biological unit. This class includes not only viruses, but also prions and satellites and viroids. Hop over here again. You can see virus. This is a subclass of acellular structure. VIDO populates the children of acellular structure by importing from the NCBI taxon when it's possible. However, terms imported needed to be supplemented with textual and logical definitions. Now, with respect to the class virus, VIDO arranges subclasses according to the Baltimore classification, which is widely used in virology circles as a way to exhaustively categorize viruses. As you can see here, this is a representation of VIDO in Protégé. Here's some terms that are associated with the Baltimore classification, which was imported. Fortunately, we were able to pull them these terms directly from the NCBI taxon. For instance, double-stranded DNA retrovirus. Over here, this smaller screen with the squiggly lines, this is a textbook description of the information that the Baltimore classification contains. Roughly speaking, the Baltimore classification is based on the genetic material of viruses and how many steps, you might say, a given virus has to take during replication process. So for instance, your coronavirus is right under retrovirus. The coronavirus is being positive sense single-stranded RNA viruses. And back to the diagram. We'll move over to pathogenic disposition, the very important class. VIDO imports from IDO Core this definition, pathogenic disposition. I'll walk through some of it. Disposition, of course, as we all know, has a material base and a material entity. Manifests in various ways. Salt is a paradigmatic example. It has the disposition to dissolve. It does not have to. A piece of salt could go on for quite some time without ever dissolving. Nevertheless, it has the disposition to dissolve if it comes in the right environment. A pathogenic disposition, it's one born by a material entity to establish localization in or produce toxins that can be transmitted to an organism. we've included both of these disjuncts here because are not all things that are called pathogens in the literature just establish localization. Clostridium botulinum, for instance, produces toxins that are transmitted or eaten or ingested by organisms, leading to disease. It itself does not often establish localization in organisms, although, of course, there are cases in which it does, namely infants, if it's tagging along with, say, honey. In any event, we want to allow for both of these cases because the term pathogen and pathogenic disposition seems to apply to a wide range of entities. We also want to leave open that a given pathogen need not cause or form disorder. HIV may be transmitted to somebody or someone who has a certain gene that protects it from forming a disorder when infected. Nevertheless, HIV can be transmitted to other immunocompetent members of the organism's species, and in any event, we would want to call HIV in those cases a pathogen. Moving over here to a subclass of pathogenic disposition, namely infectious disposition. Here we have a pathogenic disposition that's borne by a pathogen to be transmitted to a host and then become part of the infection. It's not enough for an organism just to be disposed to be transmitted to a host. That might count too many entities as infectious. It also has to be disposed to become part of an infection, so form infection in the host if it's transmitted. Now, as I mentioned, we have this immunocompetent members of the same species as host clause. HIV is a virus, HIV is infectious, and we want to capture it as such. So we do have to add this additional clause for infectious disposition as well as for pathogenic disposition. It's worth pointing out that pathogen, as we're treating it, is indexed to stages of species. Trees have pathogens, for instance, that are not pathogens for humans. Young trees, for instance, have pathogens that are not pathogens for older trees or more developed trees. And on our account, pathogens bear dispositions to localize and cause disorder. But in addition, when those pathogens are disposed to transmit and cause infection, we count them as infectious. Acellular structures bearing such an infectious disposition are what we call infectious structures. And in that class, we find quite a few viruses. Now, again, that was just some highlights of the Virus Infectious Disease Ontology. We'll hop over here to the COVID-19 Infectious Disease Ontology, the development of which was motivated by, of course, the present pandemic. And we will cover epidemiological pathogenesis and epidemic to pandemic aspects. Here you see a different diagram, also illustrating a wide range of classes from various ontologies, not just from IDO-COVID-19, but VIDO, CIDO, gene ontology, and IDO Core. We'll talk through several of these. Note these: SARS-CoV-2 disorder, and COVID-19, a disease, and a COVID-19 disease course. So a disease, and we're following the Infectious Disease Ontology core here, a disease is a disposition to manifest various symptoms and signs. For example, COVID-19 is a disease to manifest signs and symptoms that have been associated with SARS-CoV-2 infectious disorders. And any instance of COVID-19 has this disorder as a material base and may manifest in COVID-19 disease course. We represent various aspects of the disease course by importing this term pathogenesis, which we have pulled in from the gene ontology and supplemented the definition. Collections of specific processes that generate the ability of a pathogen to induce an abnormal or generally detrimental state in an organism. We can extend this further by introducing subclass for SARS-CoV-2. Here are some aspects we identify and keep clear in the ontology associated with SARS-CoV-2 transmission process, establishment of localization in a host, establishing an infection, and the appearance of disorder. And these should sound familiar. We've seen these before when we were discussing pathogenic disposition and infectious disposition. It's also worth noting that these aspects are considered parts of SARS-CoV-2 pathogenesis. Moving right along, these subclasses of viral generative stages. These are virus attachment stage, virus penetration stage. There are actually many more, especially if we're thinking about COVID-19 disease course, which has as its part SARS-CoV-2 replication. Replication, you can see it can be divided into parts that also bear precedence relation. And this is sort of straight out of virology texts, right? You see there are various stages for a given viral replication. Several of these are the targets of lots of investigations by researchers when they're trying to figure out ways to to develop vaccines to prevent virus replication. So if we can stop, for instance, an attachment stage, we can potentially drop the rate of replication of the virus in a host. We also want to provide space to represent a shift from epidemics to pandemics. And in this respect, IDO and also IDO COVID-19 have virus-specific epidemic, virus-specific pandemic terms. We're treating viral disease epidemic as a process, an infectious disease epidemic of viral disease realizations in which there is a statistically significant increase in the infectious disease incidence in a given population. So note there is a reference here to change over time. Similarly, for pandemic, we have another subclass, the subclass of infectious disease, pandemic. And here we have viral disease epidemics, so reference to the previous definition, of the same type of viral disease unfolding over overlapping periods of time and affecting populations located in different geographical regions. There's some visual representation. We have an infectious disease incidence. We could have a viral incidence right here, represented in one area. This would be a measurement of the population, as it were. Only a few infected individuals in this first globe. As we progress over time, more and more people are infected to the point in which it becomes an epidemic. Now the entire region, as you can see here, is red. Significant infection across the board in the area. And if this happens in multiple places, we have a pandemic on our hands. And a pandemic can shift while still maintaining its status as a pandemic. Different parts of the globe can be infected. Some can do better than others, until ultimately the disease spread is under control and we're over the pandemic. Some of the tracking going on here, we leverage resources from the basic formal ontology, and specifically process profiles. Typical example here would be my body temperature over time. There are lots of different processes going on in my body at any given time. I have a temperature. It fluctuates over time, as does everyone else's. Instead of focusing on the homogeneity of my system in its entirety, we often want to abstract from that and focus on some smaller subpart. We call this a process profile, so you focus on my temperature changes over time to track that. Now, here we have viral disease incidence profile. So an infectious disease incidence profile comprised of a series of determinate viral disease incidents. They're caused by a specific virus in a population over time. This abstraction is just one among many. We might also, for instance, want to look at the viral disease proportion profile, which is an infectious disease proportion profile comprised of a series of viral disease incidence proportion qualities caused by a specific virus per unit time. So we can also have rates, we can have seasonal changes. Process profiles allow us to capture that sort of epidemiological information with a very clear set of terms. Now, both VIDO and IDO COVID-19 are dealing with a rapidly evolving scientific data. We look forward to collaborating and have on several occasions collaborated with others, notably with our colleagues working on the Coronavirus Infectious Disease Ontology, of which there's significant overlap. Researchers in virology, immunology, etc., they have a hard task ahead of them wrangling this novel pathogen. We want to make the recipe for doing this sort of ontological modeling easy enough to understand. Researchers following in these steps don't have to think too much about it, because there are many other things that they should be thinking about. Thank you so much.